So to begin this, let's have a little context and define what is sustainability, right? We know there are many different definitions of sustainability, and here I'm preaching to the choir because this is the Asian School of Environment and the Earth Observatory of Singapore, right? But the most widely used definition of sustainability is this one: the development that meets the needs of the present without compromising the ability of future generations to meet their own needs. So after knowing what the definition of sustainability is, the lingering question that follows is, what are actually the needs of the present, right? So we're putting things into context. So anybody heard what the Earth Overshoot Day is? Some of you have, right? So is the day that marks the date when humanity in a given year has used all the biological resources that the planet could generate during that year. So you know what date it was for this year, or I already say it was. So you know it's been in the past. Anybody? It's been July 28th. So about a month and a half ago, we have already consumed all the biological resources that the Earth could generate for this given year. So that's our needs. As of this year, we need 1.75 Earths to meet the needs of the present, which tells us that actually the world is not enough for us, right? So, and this has been happening for a while. So this is the Earth Overshoot Day since 1970. And then ideally we will have things, you can see the cursor here, right? Ideally we will be on December 31st, if at least we could consume everything we need by the end of the year, so the Earth actually has time to generate resources for us. But the trend is getting worse. So we see this red bar overtaking the green bar little by little. And the past decade, we're moving from August all the way into July, right? And then this number, right, takes into account the planet biocapacity, which is the amount of ecological resources that the Earth is able to generate that year, and then the ecological footprint, which is the humanity's demand in terms of resources and also generation of waste for that particular year. So then the world is actually not enough for our current needs, right? We are over-exploiting resources, over-consuming products, over-producing them, over-trading. We're generating a lot of waste, generating environmental pollution, depleting our resources. The lifestyle has really changed since the Industrial Revolution. And there's a concept of market failure, which is that very cheap gadget that you paid for, right? Actually, that price probably didn't account for all these hidden environmental and social costs related to how the resources to make that gadget were generated, in which conditions the labor that actually the assembly of it worked, etc. So this is a market failure because it's a suboptimal solution, only concealing the economical aspects. So the way we were using right for the Earth Overshoot Day, it takes into account this ecological footprint, which takes into account consumption of resources and also generation of waste. And then we can see how many Earths are required to keep up with the lifestyles of different countries. For example, this is a list having some countries. Some of you here in the audience are from some of these countries. And the way this equation is calculated, right? So if in the case of the USA, their ecological footprint is 8.1 global hectares per person, and then the global biocapacity is actually 1.6 global hectares per person. So if everybody were to live the lifestyle that people in the US normally live, we would need 5.1 Earths. So I am from Argentina, so I want to see how Argentina was doing in this list. And here it is at 2.1. Also, the world is not enough for Argentinians and their lifestyle. Any guess where Singapore would be in this list? Any number? 3. So 3 is a reasonable guess. Here in Singapore, we are at 3.7. So to sustain the way we live our lives, we need 3.7 Earths. And this is the list of the top 25 countries. So Singapore is at the position number 23. And then we see the place where the World Cup is happening very soon. Requires 9 Earths to sustain that type of lifestyle. So you can have a view, again, we're putting things into context, right? Why we need sustainability. So we can see this from the point of view of countries, right? So how the citizens of a certain country live their life and what's the biocapacity of that particular country, right? So if you have a lot of desert, for example, you don't have much of biocapacity. So the ecological footprint for Japan, that is first in this list, is about 4.6 global hectares per person. And then their biocapacity is only 0.6. So that's why Japanese need actually 7.8 Japans in terms of their biocapacity to meet their actual needs. So again, you have different countries here. The case of Argentina is interesting because Argentina is a very big country that is very rich in resources, right? And has a very big biocapacity. 8th in the world in terms of surface, has only 45 million people, but produces food for 300 million people. So Argentinians, the way they live their lives, they are using half of their biocapacity. What do you think Singapore? The number for Singapore is going to be? Any guess what? Very high, huh? No, anybody wants to guess a number? Huh? 8, 20. I got 20. Anybody higher than 20? 50.62. All right. Anybody higher than that? So you'd be surprised. It is 104.4. So people, us living in Singapore, we consume, 104.4 more times the biocapacity of this country with our lifestyles. And if you take the top 25 countries in the world according to this criteria, Singapore wins by far. I was surprised. I didn't make up these numbers. So yeah, Singapore number 1. So really food for thought. It's related, of course, with many factors: the biocapacity of this land, how many people are here, and the lifestyle we have. But it's very good to get us thinking and to put us into action, because if that is the situation, right? So the needs of the present are too high, then we cannot really be sustainable thinking into the future. So then the path to sustainable development, a really good one, has been actually set up in 2015. So that's 7 years ago by the United Nations with the goals for sustainable development, right? These 17 goals that are an urgent call for action by all countries in a global partnership, right? So again, preaching to the choir here, but it's important to know that all countries have to work at the same time, because if one country only focuses on carbon emissions and the other one doesn't reduce them, then it doesn't work. We all have the same planet. Because we share the planet, this is a shared blueprint for prosperity, people now into the future, and the planet. So something I want to share with you, which you might have not seen, is this representation, which is the wedding cake of the SDGs. Anybody has seen it before? There you go. So something new to learn. So this is from the Stockholm Resilience Center. Actually has been done in 2016. And it really takes into account that economies and societies should be seen as embedded parts of the biosphere. So some of the SDGs are related to the biosphere primarily, some to the society, some to the economy. And then the icing on the cake is the partnerships for the goals, because we all have to work together. But of course, the SDGs are all intertwined. So different SDGs impact others. And that means that the way organizations work, right, we're shifting, or we have to shift what we call normally the bottom line in business jargon, which is what's the bottom line money? Well, it's not money only anymore. Now we also have to take into account the people and the planet and the intertwined relations that we have between these two aspects and profit. So if you want to know where we are actually making progress since the SDGs were released, there are sources to do that. I also want to share with you a couple of TED Talks. This one is from 2015, presenting the SDGs, and this one is from 2018, 3 years after. They use the Social Progress Index in order to measure how different countries are doing in terms of SDGs. Very interesting talk. And then each year, the UN releases a Sustainable Development Goals report. So there's the one for this year already released. And it's not so encouraging. We still have a lot to do. And the pandemic was a hard strike on many different aspects that we were making progress, or us as mankind. But then we need to work on it in order to get into pace again to achieving them by 2030. So you can check this out later in your time. And then Singapore actually last year released the Singapore Green Plan, which is an effort. I'm going to read this because I like it. A national sustainability movement which seeks to rally bold and collective action to tackle climate change. But if you look at it, it's not only climate change, it's actually different aspects of the Sustainable Development Goals. Climate change is only one of them. So these are different pillars of the SDG Green Plan: city nature, sustainable living, a resilient future, energy reset, and green and economy and green government. And then many of us here in this audience, online audience too, you're probably working to try to tackle many of these aspects of the SDG Green Plan and the SDGs. Particularly our research focus and the research I work on focuses on sustainable living and a resilient future, which in turn is connected to a green economy. So this is all important because we actually have to transition from the traditional linear economy that comes from the times of the Industrial Revolution, that considers that we have infinite resources, which we can just extract, make something, and then after the lifespan of that product, dispose it as if we had an infinite capacity of regeneration of the planet, towards a circular economy where everything in the value chain for this product is considered, from how the resources are going to be extracted, what type of energy is going to be sourced, and then the design of the product actually accounts for later on being able to recover, reuse, and recycle. So something that I want to recommend you guys to read if you're interested in this is the Cradle to Cradle book in 2000. It's actually from 2002. And this book has changed the way I see many aspects of sustainability. I used it in a class of green engineering for sustainable design that I was at TA and at UC Davis. And the book itself is not made of paper. It's made of recycled plastic. The first chapter is called "This Book is Not a Tree." And it really shows that to go to a circular economy, we have to start in the design, right? So if you take your phone and then there's no way for you to open it to change parts of it, then the design is made in a way that we can actually not recover those valuable materials, right? So the design is very important, and it takes a concept that we have to mimic nature where waste equals food. So there's really no waste. So just wanted to share with you guys this case for the case of food waste, our very own TEDx NTU talk in 2018. It's called the Food Waste Dilemma. Some of you, I think, were there in person. A third of the food that we generate globally goes to waste directly. And he presents some of the reasons why. This guy is a freegan, which means that he tries to live for free. At the time of the talk, he told us that one year he lived on only $8 for the full year. And from those $8, $5 he spent on buying a coffee to his crush. So then he literally is able to live for free by eating perfectly edible food that we normally waste. So I invite you to check that out. And then wanted to share with you this resource from the Ellen MacArthur Foundation, which is also a very important resource if you want to learn more about circular economy, that shows this butterfly diagram. It shows the cycle of renewables, which is the biological cycle on the left, and then the finite materials, which is the technical cycle on the right. And then how, in the case of renewables, nutrients from biodegradable materials can be returned to Earth through different processes. Well, this in the technical cycle, materials are kept in circulation through cycles of reuse, repair, remanufacture, and recycling. But for this, it's very important to design this from the very beginning, so we can facilitate all the value chain for this to actually happen. So this was some context, and now we're getting to the microbes part of the talk because of their role that they play in this biological cycle, right? And I work at CELSI, the Singapore Center for Environmental Life Sciences Engineering, as many people here in the audience. And there we think that biofilms are ingrained in all aspects of life, right? So they are typically here highly complex and diverse microbial consortia. Microbes are all these tiny creatures that are invisible in naked eye, but we can often see them with a microscope, and they comprise all domains of life. And they are all over our lives, right? They can cycle, they have an immense metabolic capacity. For example, we need oxygen to be alive. Many microbes don't need oxygen to be alive. They cycle important nutrients in important atoms in the planet, like the cycle of carbon, the cycle of nitrogen, the cycle of sulfur, phosphorus. They're present in all sorts of ecosystems. They can help us produce food. Think of cheese, think of yogurt. They're important in the urban water cycle. We can use them to bioremediate pollutants. They are involved in diseases, like COVID is a virus. Actually, SARS-CoV-2 is a virus. And they're also important as part of host health and well-being. So we are holobionts. Did you know that we have more bacterial cells in our body than cells of ourselves? The ratio is actually 10:1, is estimated. So there are more bacterial cells in you than there are cells of you. And they are also important in aspects like some of you guys working here, biofouling, biofouling, biocorrosion. So literally, because of their broad spectrum, they're involved in one way or another with all the different aspects of sustainable development that we want to tackle. And I want to show an example of something that I work on a lot, which is water treatment. So here you see this video. I hope you guys in the online audience can see it too. This video, we see bacterial flocks, which are these clouds, and then we see some protozoans grazing in the background. And this is what you see here in a sample of this biological wastewater treatment tank. And it's a complex microbial system that has hundreds of thousands of different taxa, thousands of genes, trillions of individuals. This is a very interesting concept. So if you think about it, right, 1 milliliter of sludge will have 1 billion cells. And these are all individual organisms, right? So you have 8 milliliters, you have already more individual organisms than the population of humans on Earth, right? So this is, again, each cell is an individual organism. So, and the reason why they are there is because they are eating. For our waste, for them, is their food. So this is the principle behind this. And this system serves a major role in the protection of water quality and public health, but it's also a renewable source of fresh water, and which is very important in the case of Singapore, right? The objective by 2060 is to have treated sewage meeting 50% of the water demand, right? And some people think of the yak factor. Stefan likes to mention that. But then in fairness, recycling the wastewater is basically mimicking what nature does. And we're humans, we're part of nature too. So another way of seeing this is to see, okay, waste. So the paradigm for wastewater treatment is changing from just treating waste to reduce amount of nutrients and pathogens in the water to actually recovering resources. So the raw wastewater enters into a resource recovery factory where we can recover water, we can generate nutrients, we can recover nitrogen, we can recover phosphorus, we can even recover microbial protein, and then we can also use these cells through digestion to recover energy by the generation of methane or hydrogen. Again, helping achieve many of the sustainable development goals. We can also track the presence of viruses in the wastewater, like what Janelle does and her team. So there are many ways in which this system serves as an example of how we can tackle the SDGs. So then within this context, I just want to share with you 3 examples of the research that I work on, where we think we're moving towards sustainability through the ecology and engineering of microbial systems, because since the spectrum of things that microbes can do is very broad, you need actually multiple disciplines in order to be able to understand and really harness these microbial communities. So not only microbiology, but also ecology, engineering, data science, biotechnology, and so forth. So the first case is related to soil bioremediation of oil-contaminated soil. So this is a project that I did with an industrial collaborator. This is Dr. Poi. And then what we wanted to do here is to tackle the problem of oil waste that happens worldwide, but also here in Singapore, where the oil industry, there are refineries, you probably seen the flame from the beach, right? And it's a very important industry here, but it has a big footprint. Whenever there's an oil spill, the practice is to remove that soil that is contaminated with oil, send it for thermal desorption or incineration. And then after that, the residuals are buried in Singapore's landfill. And then on top of that, that removed soil is replaced with clean soil that normally is sourced from overseas. So think of all the emissions of carbon dioxide and the cost, the energy involved in this process. Moreover, we're running out of space in the landfill here in Singapore. The only landfill is predicted to reach its capacity by 2035. And because of that, the circular economy aspect of the SG Green Plan requires us to reduce the waste sent to the landfill per capita per day by 20% in 2026, which is not so far from now, 4 years, and by 30% in 2030. So this problem calls for a solution that is environmentally sustainable and economically feasible. And then we think that microbial remediation, bioremediation, can be that solution. And this would be the biodegradation of chemical pollutants but microorganisms, which can be often done with the aid of biostimulation, which is when you supply nutrients. And currently, there is not such technology or application here in Singapore, other than the one from my industrial collaborator. And then this market is growing and expected to continue to grow in the future. So it's a good opportunity, not only for the sake of a better planet, but also from the environmental, economical point of view. And something that I forgot to mention about circularity is that being optimal or optimizing all your flows is actually better. Not only helps you save money, but also improve the way you impact the environment for the better. So to grow microbial cultures, in order to do bioremediation of oil contaminated soil, you need some means to do that, right? Some sort of vessel. So traditionally, traditional microbial fermenters are good. They serve the purpose, but they have low flexibility. They're expensive and have a high carbon footprint. There are also bags that are single-use, developed for mammalian cells growth, which are also more expensive, not as expensive as the previous one, but still expensive, and they have a medium carbon footprint overall in comparison. And then what we propose is to combine ideas from these two types of technologies and then add some elements of our own in order to come with a multiple-use bag bioreactor system where we could grow cultures fit for purpose depending on the demands of oil bioremediation there in a way that is modular, easy to scale up, and at a lower carbon footprint. So we did that. So here you see an example of these bags growing cultures. We're growing a proprietary microbial consortia that we know it's able to perform this bioremediation, and then it's growing these bags in multiple cycles. And we tested the suitability for the bioremediation in the field in a pilot patch here in Singapore by deploying these microbial cultures and tracking the removal of total petroleum hydrocarbons over time. So we can see that about 76% of it was removed over 50 days, 53 days, and then 79% over 90 days. So this shows that a lot of the contamination can actually be removed through bioremediation in this particular pilot trial in Singapore, but there have been others where actually you can reach to 100%. And then we also assessed the microbial communities, how they changed within these soil samples over time. And then we could see that actually there was a shift in their composition and relative abundances and some interesting dynamics of bacterial diversity, starting with a higher diversity, then decreasing. And then when a different set of microbes takes over, diversity increasing again, but then decreasing towards the end of the experiment. So overall, this was interesting because this proposal tries to shift us from the current expensive and unsustainable way of handling these oil contaminated soils. Look at all these costs over here for all the different aspects involved, into something that's more cost effective, but also sustainable, helping reduce oil pollution, lowering our carbon footprint, reducing landfill use, and then recovering soil so you can reuse it again. So bioremediation can help us achieve these different SDGs. So this is just a small acknowledgement picture to acknowledge members of the team. We had some wonderful interns that helped us with wet lab work, with the design and testing of the prototypes of the bioreactors and the molecular work. So the second example is a way bigger project. That's why I have the logos of many agencies that also work with us, which is related to the production of microbial protein for food processing wastewaters as aquaculture feed additives. So here's Beta over here, which is there in the audience. And then the rationale behind this project, right, is that the world population is growing and we need to feed this population growth, right? And that requires more resources. And already as of today, we need 1.75 planets, remember? So we are not doing well in that regard. So the fastest animal protein sector is aquaculture, the fastest growing animal protein sector. And then the way aquaculture, the fish are fed is traditional fish meal is actually sustainable. It has a lot of fish. So we need sustainable animal feed resources, alternative ones, in order to feed fish for aquaculture. So we put together this project. I mean, Stefan Wertz, who's here in the audience, he's the lead PI, in order to try to recycle food processing wastewaters, which are rich in nutrients, to provide high quality aquaculture feed. And this is very relevant for Singapore because there are over 100 fish farms in Singapore and they only meet 10% of the local consumption, right? Which means that there's room for aquaculture to grow in Singapore and it's actually growing. And then we also have some industries that are food or beverage processing that have nutrient-rich wastewaters that are currently just discharged and treated in the wastewater treatment plant. So not resource recovery, but the old way, which is just wastewater treatment. So here we started with this project, and then here Beta, who's here in the audience, he wrote this very nice review today with me and Stefan. And this was very interesting because when Beta started his PhD and we were working in the lab, and then we had to start trying to convert these food processing wastewaters into protein, the first thing you do is you don't try to reinvent the wheel, you go and check what's there in the literature, right? So I remember asking Beta to do that, then he came back and he had very few papers, and I said, like, Beta, you didn't search strong enough, like you didn't search hard enough. This cannot be. So after, you know, this conversation, a friendly conversation, then he went, search harder, and only find a few more papers, right? So really there was nobody actually trying to use a microbial community-based protein production approach from food processing wastewaters, from animal feed applications. And he had to change it to wastewater in order to accommodate more papers because so few pieces of work were addressing food processing wastewaters. So I remember the previous seminar from Professor Young when he talked about a review that one of his students wrote, and now it has 3,000 citations. So I hope you get lucky with your review. Fingers crossed. Yeah, I want to share this. So it's been a pleasure to mentor Beta in his journey through his PhD. So the highlights of this work is that food processing wastewaters actually can support microbial growth and protein synthesis, but then very few works actually do deep chemical and microbial characterizations from these food processing wastewaters. So then part of this work, there's this figure where we show that the animal requirement of certain essential amino acids, which means that the fish cannot produce it by themselves. They need to take it in their food. This is a requirement for trout and shrimp. And then this microbial protein produced according to the references by different sources of food processing wastewaters. And then you see that many of these essential amino acids, more or way more than the requirement, is produced from that, is available in this microbial protein. So then what we did is to go and try to grow some of this microbial protein ourselves. So we took some wastewaters from the soybean processing industry here in Singapore that has some valuable nutrients. We assessed its microbial composition, and then we found that there in that community, there were many microorganisms that have probiotic potential, like Lactococcus, Lactobacillus, Weissella. And all this work is actually available if you want to have a look on a preprint, where Beta is again the first author. And then basically what we did was to basically take these food processing wastewaters using those microbes that were there present, and then provide conditions in the lab for fermentation, and then basically grow this microbial protein that then we dried. And then we checked its amino acids composition and compared that with the animal requirements for trout and shrimp. And then we saw that actually it was very good. So here we pasted a label in our microbial protein bottle in order to see the nutritional composition of it. But then we needed to know whether actually the fish would like to eat this, right? Maybe it wouldn't be palatable for the fish. Maybe they wouldn't eat it at all, right? So then with our friends at the Aquaculture Innovation Center, we did one trial so far where we took Asian sea bass, and then we formulated two different diets. When one had 30% of the protein composition, 50% of the protein content that comes from fish meal replaced by single-cell protein produced in our lab. And then this is with Beta when we brought because we helped them bring the fish from St. John Island. And then these are the fish actually eating the experimental diet. So it was very nice for us to see that the fish actually ate the microbial protein we grew in the lab. Of course, through a feed formulation mixed with other components, right? But 50% of the protein from that came actually from our microbial protein grown in the lab. And this was very, very, very good because when we checked the weight gain of this fish, we saw that it wasn't significantly different from that of a normal fish meal group. And then moreover, it was actually less variable, which suggests that maybe using single-cell protein could lead to a more homogeneous growth of the fish. Other parameters like food conversion ratio or specific growth rate were also not statistically different. But again, we see less variability for the case of the experimental diet that has the single-cell protein. And then this, we have a manuscript that hopefully we'll submit very soon, and then we're going to be presenting on the World Aquaculture Singapore conference in November. So this is great because this can actually help us achieve all these different SDGs, zero hunger by producing alternative sources of protein, and also clean water sanitation, because we're taking that wastewater, transforming it from a waste into a raw material to help having more sustainable cities and communities and more responsible consumption and production. And this is a bonus. So I'm having a new hobby, which is ant keeping. What time is it? Okay, 1:36. So we have a nice TED talk, TEDx NTU talk in 2019 from an ant keeper, which I recommend you to check. And then to grow ants, you need sources of protein and sources of carbohydrates, right? Carbohydrates for energy and then protein to growth. So I talked to Beta, like maybe we can a little bit of powder because they don't really need much as a source of protein, and then we see how it goes. Otherwise, I have to feed them bugs, which is what they normally grow, right? And then this is a very starting colony, the very first one I have. This is the queen with 3 ants. And then here it has 4 ants and then some eggs and then a little bit less of what I initially put. And now there are 8 ants. This is of today, this morning. There's very few protein left. This is not a real experiment, but I thought it's fun. Like they are actually growing, and the only protein available here is coming from the microbial protein. So it's good. Now we have an alternative for for ant keeping. Yeah. So it's really nice to have these pets. So this is a massive project. I mean, ProfWorks is the lead PI, but we have collaborations with many different people, many different co-PIs, and then I only show you one aspect of this project. There's also a sustainability and economic feasibility assessment aspect of this project, and we're only halfway into the project. So it's very exciting and very nice to be a part of it. And this is how big the team is. Again, different co-PIs, but also different research fellows, PhD students, RAs, and then we're partnering with a lot of, with several local food and beverage processing companies, as well as aquaculture companies. So again, really nice project to work on, and then we'll see how we do in the next couple of years. And then this is the last case that I want to share with you. So I'm putting this picture here because once I wanted to show you that once I had this beautiful beard. Stefan, you also had a beard. We both shaved. And then a lot of this comes from my PhD work. So Stefan was my PhD supervisor. I also worked on it as part of my research fellow work. I continue to work on it, albeit slowly than before. And then in this particular case, we take a more broader perspective to use microbial systems as model systems for asking questions related to community ecology, right? And particularly to disturbance. And why do we do that? Because disturbance events are increasing and changing worldwide. And this is related, according to many experts, to our impact, right, to the population growth. So we're having higher frequency and intensity and variety of disturbance events worldwide. It is well documented. So this is a paper from Prof. Horton, the director of EOS, where we see there's well-documented increase of carbon dioxide emissions, terrestrial biosphere degradation, increasing global sea level, the global temperature, and all these things related to human activity are actually related to an increase in disturbances. So here we have, for example, storms, floods, landslides, avalanches, heat waves, droughts, fires, right? So this is happening right now. As a consequence, we are potentially seeing the 6th mass extinction event, right? So there's no yet consensus that this is actually happening right now, but many people do believe it is. So these are the actual rates of extinction of many different species, and this is related to our impact in the world. So then understanding the effect of disturbance in communities is more relevant than ever, right? And then disturbance is important because it structures the ecosystems. It changes the composition and relative abundances of its members. And although it can be defined as an event that changes resources, social availability, and the physical environment, it can also be an event that physically injures or kills or inhibits members of a community, but at the same time creates opportunities for others to grow and reproduce. So for example, succession, which is the sequential replacement of the species, often occurs after a disturbance like a fire in this particular example. And disturbance is also important because it can lead systems, or in terms of their structure or their ecosystem functions, into alternative states, alternative stable states that may or may not be reversible. So I showed you this, but this is a very nice system to ask questions about disturbance ecology, because it's a very complex system, and also because we can track ecosystem functions very easily in this system. And then also most bacteria in activity slash have generation times of less than 24 hours. So reflect of it, especially some in the audience are ecologists, right? Reflect of it for a second, because 24 hours, right, that's the slowest bacteria in terms of the generations. And that would be in a forest equivalent to the oldest trees that takes many years, right? And then we have bacteria that have doubling times of a few minutes or a few hours, right? So then we can really ask questions in a shorter time span in terms of successional dynamics. And then try to see where we can actually draw generalized conclusions through the ecological theory from specific observations of these organisms in their environment in order to make predictions about the world around us, right? So this is the whole goal. And I'm an engineer, so of course I don't know much ecology as the actual ecologists in the audience, but I really like this idea, and comes back to tie with the multidisciplinarity approach that I was talking to you earlier. So in Celsius, we worked with different types and scales of disturbances. The work I did with aerobic reactors here. One type of disturbance was symbiotic trichloroaniline, which is a compound that is a runoff from the use of pesticides and herbicides in agriculture. And the other one is organic loading shocks, here in the form of double COD, which is something that you would have, for example, in wastewater treatment plants of food processing companies, right? A dairy industry or something like that. And then we worked at different levels in terms of volume, scale of volume and time. So microcosm level and shortened time spans of about a few weeks. And also at higher, two orders of magnitude higher levels of volume, mesocosm, and also longer terms in terms of days that the experiments were run. And then we did this because disturbance is a multidimensional concept. It can vary in type, frequency, intensity, and its effects can be uncertain. So that's why we need variability. So in order to capture that variability, we need replicated systems, because remember, disturbance can lead the system to alternative stable states that may or may not be reversible. So then for this case, just focus on this particular microcosm studies because we don't have much time, but we wanted to ask what happens when we subject these reactors to different frequencies of disturbance. And then one theory from ecology, very old one from 78, is the intermediate disturbance hypothesis that predicts a peak in diversity at intermediate levels of disturbance due to competition colonization trade-off that species face. And this is a nice theory and many people have tested it, but the problem that gaps that it has is that it only takes into account the richness portion of diversity, which is, okay, what species are there, but it doesn't take into account relative abundances, and also leaves gaps related to the role of assembly mechanisms, either deterministic or stochastic, and then what, if any, would be the relationship with functions in the systems. So what we studied this using a microcosm system with trichloroaniline as the disturbance, dosified at different frequencies, and then we observed that there was a peak in alpha diversity intermediate levels, but then we hypothesized that this was related to the low predictability of this environment that we find at intermediate levels of disturbance, right? So that will favor stochastic processes that in turn will select for a higher diverse cohort of microorganisms, whereas extreme size of the disturbance range, where conditions are more predictable because they're more recurrent, will have a stronger role of deterministic processes, and that would lead to a few species dominating, and because of that, we will have lower alpha diversity. So because this was done with only one type of disturbance, we tried again with another type of disturbance, the double organic loading, and then we saw that with time, there was a peak in alpha diversity, again, so this is a metric of alpha diversity, second order Hill number, takes into account composition and relative abundances as well. And then we saw the development of a peak in alpha diversity as predicted by the intermediate disturbance hypothesis, but as predicted by our intermediate stochasticity hypothesis, we also have a peak in the contribution of stochastic mechanisms of assembly. So here, this is the nearest taxon index, and the closer it is to zero, the higher the contribution of stochastic assembly mechanisms under the particular null model that this methodology uses. And then something very interesting as well was that this peak preceded the formation of a peak in diversity, right? So assembly was preceding structure, which is something very interesting to see. And that's why we thought these findings were very exciting, because we saw that assembly actually preceded structure. And then it could be that stochastic mechanisms operating at intermediate levels of disturbance could be the reason why sometimes a more diverse system does not translate into a better function. And then we put together a framework to try to explain that. And if you're interested, please check this paper where we describe it. And then better than that is to try and check this blog that I wrote to try to explain what we did in this work, but more to a general audience, and how we think we can use these tools to try to anticipate changes in biodiversity and an increasingly disturbed planet. So we did more work related to disturbance ecology, and all of this is related to several of SDGs. But in the interest of time, I cannot show it. But if you want, later you can talk to me or send me an email or have a look at these works. And then I also want to acknowledge a lot of people who helped in this particular case for the disturbance ecology portion, particularly my PI, Prof. Wertz, but also Florentine Constanzas, which helped me a lot with metagenomics, bioinformatics, and Harry's session. And then this work is ongoing now. Sohail, who is here in the audience, Avid, with Stefan, are taking it to test these ideas in anaerobic systems to see the wider applicability of them. But we contend they should be also applicable to other systems, but that's for everybody to try. So this brings me to the end, and then we'll have 12 minutes for a discussion. So I hope that we could see that we can move towards sustainability through the ecology and engineering of microbial systems. And for that, we really need to harness microbial systems, and it requires a multidisciplinary approach. And then I just want to leave you with this slide so you get to remember how microbes are everywhere. Thanks.